A Greenland shark swimming in the North Atlantic right now could have been alive before the United States existed as a country. Before the Eiffel Tower, before the printing press reached most of Europe. Current research estimates these animals can live somewhere between 200 and 500 years, making them, by a wide margin, the longest-lived vertebrate on the entire planet. Meanwhile, a mayfly completes its adult existence within 24 hours. Same planet, same basic biological building blocks.
So what is actually going on here? Why did evolution decide some animals deserve centuries?
Let's start with the single biggest, most consistent factor explaining longevity differences across the animal kingdom: body size. Specifically, the relationship between metabolic rate and lifespan. Larger animals tend to live longer than smaller ones. This comes down to the rate of living theory. Animals with faster metabolisms age and wear out faster.
A mouse has a frantic metabolism, its heart racing, constantly burning energy to maintain body temperature.
An elephant, by contrast, operates with a considerably slower, more measured metabolic rate relative to its enormous size. Elephants can live 60 to 70 years, a staggering difference compared to a mouse's typical 1 to 3 year lifespan. This relationship isn't perfectly linear. If body size alone explained everything, we wouldn't need an entire video on this topic. There are plenty of fascinating exceptions that completely break this simple pattern. Let's bring up BATS. BATS represent one of the most striking exceptions to the basic size longevity pattern. BATs are small, have fast metabolisms, similar to small rodents, yet can live 30, even 40 years, dramatically outliving similarly sized mammals. Researchers studying this discrepancy have proposed several explanations. BATS fly, an activity demanding sophisticated cellular damage repair mechanisms compared to ground-dwelling animals of similar size. The physical demands and oxidative stress of flight suggest BATs evolved robust cellular repair and stress-resistant systems. These repair mechanisms provide protective benefits against general aging, a side effect of evolving to survive flight stress. This extended overall lifespan beyond what their small size and fast metabolism would predict. Now, let's discuss naked mole rats. Naked Molder rats are small, rodent-sized animals. Based on size and metabolism, they should live 2 to 3 years. Instead, naked mole rats can live over 30 years, a bizarre outlier making them intensely studied in aging research. Naked mole rats show little age-related decline, maintaining health and bone density throughout their long lives. Unlike most mammals, naked molar rats don't deteriorate. Researchers found biological features contributing to their aging resistance. Naked Molder rats have efficient cellular damage repair and unusually low cancer rates, counterintuitive for their long lifespan. More time means more cellular mutations. Naked rats have genetic adaptations for oxidative stress and protein quality control. Naked rats show little age-related decline, maintaining health and bone density throughout their long lives. Cellular metabolism produces free radicals, which cause cumulative damage to cells, proteins, and DNA if not neutralized. Animals with faster metabolisms produce more damaging free radicals, explaining why small-bodied animals age quickly. Animals like BATs and naked mole rats break this pattern by evolving effective methods to repair oxidative damage. These animals don't just have low metabolic rates. Now, let's discuss telomeres, a frequently discussed mechanism in longevity. Telomeres are protective caps on chromosomes that shorten with each cell division, eventually triggering cellular aging or death. A popular assumption is that telomere length alone determines lifespan. Longer telomeres equal longer life. Actual research is more nuanced. Telomere dynamics are one piece of a larger, more complex aging puzzle. Several long-lived species don't have extended telomeres, suggesting they aren't the primary driver of longevity. Let's discuss predation pressure. This connects to an evolutionary theory explaining why certain species evolved longer lifespans, separate from metabolic mechanisms. Species facing high predation or environmental danger evolved toward faster reproduction and shorter lifespans. Investing in long-term cellular repair makes less sense for animals unlikely to survive long enough to benefit from it. A mouse gains more from quick reproduction than investing in energy-expensive cellular repair systems for a longer lifespan. It's statistically unlikely to ever reach. This theory explains why animals with few predators, large size, or protective features evolve longer lifespans.
Their reduced predation risk made evolutionary investment in long-term cellular maintenance and repair worthwhile for a longer lifespan.
Let's bring up tortoises now, because they provide a genuinely compelling real-world example connecting several of these threads together. Giant tortoises can live well over 100 years, combining longevity-favoring factors like large body size and slow metabolism. Tortoises live over 100 years, thanks to their hard, protective shell, which reduces predation risk. This dramatically reduces predation risk, unlike smaller, vulnerable animals. Now, how do researchers verify these animals' ages? Dating long-lived animals is a scientific challenge. For example, determining a wild Greenland shark's age required a novel dating technique. Greenland sharks lack growth rings. Researchers used radiocarbon dating on proteins in the shark's eye lens to determine its age. The shark's eye lens forms early, remaining stable, acting as a biological time capsule for radiocarbon levels. This clever, indirect scientific detective work is crucial for studying difficult questions that cannot be directly observed or measured. It's worth noting the considerable lifespan variation within individual species, not just when comparing different species. Lifespan variation exists within a single species. Diet, stress, genetics, and resources all affect lifespan meaningfully. Longevity is a complex interaction of genetic, evolutionary, and environmental factors, not a fixed number by species identity. Another interesting angle involves birds, a significant exception to the body size pattern, more so than people assume. Many parrot species can live 60, 70, even 80 years, a remarkable lifespan for a small, fast metabolism animal. Researchers link avian longevity to flight, similar to BATs, and also to brain size and cognitive complexity. Parrots and corvids, known for intelligence, are long-lived birds. Researchers propose evolutionary investment in cognition supports cellular maintenance. The causal link between intelligence and longevity is an active research area, not a settled conclusion. Now, let's discuss clams. Clams hold one of the most extreme longevity records in the animal kingdom, outside the vertebrate world. An ocean quahog clam, nicknamed Ming, was found to be approximately 500 years old, dating to the Ming dynasty. Clams achieve extreme longevity through an extraordinarily slow metabolism and a remarkably stable, low-stress ocean environment. Clams are shielded from predation and environmental volatility, unlike species pushed towards faster, shorter-lived strategies. Cellular senescence is a state where cells stop dividing but don't die, distinct from oxidative stress and telomere shortening. Senescent cells linger, releasing inflammatory signals contributing to tissue dysfunction and aging. Long-lived species prevent their accumulation. Long-lived species clear dysfunctional cells more efficiently, a cellular mechanism contributing to extended lifespan beyond basic repair. Finally, let's consider how human lifespan fits into this comparative picture. Humans are a genuinely interesting case study. Humans live considerably longer than size-based predictions suggest. Researchers studying human longevity point to several contributing factors. Our large brains, strong social cooperation, resource sharing, and extended post-reproductive lifespan contribute to human longevity. Researchers connect this to the grandmother hypothesis. Older individuals contributing resources and knowledge to family provide evolutionary advantage. This provided evolutionary advantage beyond individual reproduction. Finally, comparative longevity research is rapidly advancing. New long-lived species and mechanisms are discovered rapidly. Genetic sequencing allows comparing genomes of long-lived and shorter-lived species. Genetic sequencing provides precise, detailed insights into specific genetic adaptations driving extreme longevity across diverse species. This ongoing research is an active scientific frontier. Fundamental questions remain fascinating as detailed answers are continuously uncovered. Why do some animals live longer? The complete answer is a complex, interconnected combination of factors, not a single simple explanation.
Lifespan is a complex, interconnected combination of factors, a carefully calibrated evolutionary trade-off, not a fixed universal biological constant.